All right, so this is our first video on meiosis, which is our second type of cell division that's going to be in this unit. Our two big questions. Number one, we're going to look at how gametes, which are sperm and egg, created. Gametes, again, are a special type of cell that are only used by sexually reproducing organisms. And we are also going to look at how this process increases genetic variation between generations. So my first question for you guys is, which of the following are types of reproduction? Good, so hopefully you were able to get that the two types of reproduction, and one way that we classify organisms in biology is we have sexually reproducing organisms and we have asexually reproducing organisms. Now the interesting thing is that there are advantages and disadvantages to each. So if you're an asexually reproducing organism, an advantage is that you don't need a partner. Asexually reproducing organisms on their own are able to divide and create two new organisms. So they're able to have offspring without finding a partner. Because they are not looking for a partner, they're able to do this quite a bit more quickly. However, in asexual reproduction, remember that 100% of the genetic information from the parent is passed on to the offspring. So you get less genetic variation. Sexual reproduction, so these are organisms that require two organisms, each one contributing half of the genetic information to form a single organism. You need a partner for this. Problem with needing a partner is not only just time, it's also that it's a little bit dangerous in nature to look for a partner. So if you look out into the animal kingdom, lightning bugs light up in order to attract mates. Your frogs and a lot of other organisms, they have vocal signaling, or even birds, they have visual signals. All of these things that they're performing in nature to help them to attract a partner also attract predators. And so it makes it dangerous to go through this process and to have mating rituals. And also, typically, you have a longer generation time. When I say longer generation time, I mean that it takes longer for organisms in sexually reproducing populations in general to reach sexual maturity where they can actually have offspring of their own. The good thing, and this is a huge good thing, sexual reproduction produces a ton of genetic variation every single generation. Now, we've been talking a lot about genetic variation, and we want to kind of rewind back to evolution from the beginning of the year to remind ourselves, why do we care about genetic variation? So remember that the environment and conditions for populations are constantly changing. And if you have a population with a lot of variation in it, it greatly increases the chances that when that environment changes, there's at least one variant, so one difference in that entire population that is able to survive the change. If everybody's exactly the same, then it makes it much less likely that anyone's going to survive. So you get a lot more extinction events where everybody just dies out. So if you look down here on the bottom, the example that we have are brown bugs and green bugs. So they're showing us that the green bugs are at a disadvantage to the bird. They get eaten more quickly. Now, if we have an entire population of green bugs, then nobody has an advantage. Everybody gets eaten. Whereas here we see that evolution occurs and that this population survives because some of them are brown, because some of them are able to blend in. And this variation is what helps populations to persist for long periods of time. There are four sources of variation that we want to know about that exist in nature. Number one is mutation. Mutation is when you have random changes in the DNA that can result in new phenotypes and genotypes. So this basically means you get increased genetic variation. And this occurs in both asexually and sexually reproducing populations. So all organisms, regardless of which type they are, have mutation as a source of variation. The other three only occur in eukaryotic sexually reproducing organisms. The second one, DNA contributions from two organisms. So literally the fact that you get half your info from one parent, half your info from another, and then it mixes together to make a random new you, that is a huge source of variation. The other two we're going to look at in terms of meiosis, and these are new terms for us. Number three is what's known as Mendel's law of independent assortment. There are three laws that Gregor Mendel, who's considered the father of modern genetics, came up with. We'll go through these again in genetics, but we introduce the independent assortment here because it's easy to visualize with meiotic division. Number four is crossing over. So this is something where, again, happens during meiosis, increases genetic variation, but it only occurs in sexually reproducing organisms. When we look at genetic variation in prokaryotic reproduction, remember that prokaryotes, so your bacteria, they do what's known as binary fission, which means that essentially one bacteria copies its entire genome, splits into two. An advantage here is that they have a very short generation time, but realize that bacteria, the only source of variation between every single generation of bacteria is mutation. Now, the good thing for them is that they have a really short generation time, which means that a single bacteria in great conditions can divide once every 10 minutes, which means that in a few hours, they can create a pretty decent population. Your chance of mutation occurs whenever you copy DNA. So their chances of mutating in a shorter period of time are much higher than a human's, where it takes us 15, 20 years in order to be able to reproduce. So sexual reproduction in eukaryotes relies on meiosis. Meiosis, again, is the type of division that's only going to occur in sexually reproducing eukaryotes because it's what creates the sperm and egg cells. So it creates those gamete cells that are haploid. Remember that haploid describes a cell that only has one copy of every chromosome. So in mitosis, we were talking a lot about our diploid cells. These gametes are then going to combine during a process known as fertilization to create a brand new combination of chromosomes. This new combination means that literally every time, since you get a random copy from mom, a random copy from dad, and then you put those together to get a brand new combination that has never existed before, means that every single person and every time sexual reproduction and fertilization occurs, you get a whole new combination of genes. Which cells in sexually reproducing multicellular eukaryotic organisms perform mitosis? Good, so hopefully you were able to remember that those are your somatic cells. Somatic is just a vocab term for body cells. So your body cells are the ones that do mitosis, not your gametes. What is the end product of mitosis? Good, so what we want to end up with at the end of mitosis is 2 diploid daughter cells. So our cells at the end of mitosis should have a mom and a dad copy of every chromosome. So di meaning 2, 1 copy of each. Whereas again, in meiosis, we're going to make 4 cells that are haploid, so they only have 1 copy of each. We're going to very briefly go through the stages of meiosis to better understand independent assortment and crossing over. We'll go over this a lot more in detail in class. I just want to give you a broad overview of what happens. There are actually 2 phases to mitosis, but when we look at, or meiosis rather, but when we look at what happens before we get into meiotic division, we notice that we have interphase. And interphase is the same between mitosis and meiosis, meaning that we still have G1, S, and G2. G1 and G2 are both about growth and about copying those organelles. And S phase, S for synthesis, which means make. So what do we make in the S phase before mitosis and meiosis? We make a copy of all of the DNA. So G1S, G2, exactly the same mitosis and meiosis. Nothing changes. Our big differences before we get into mitosis and meiosis that we're going to go over. So when we divide through meiosis, there are 2 rounds of division. So how we're able to go from a diploid cell to a haploid cell in meiosis is you have 1 round of copying genetic information during S phase, but then you divide that cell 2 times instead of just once like in mitosis. So in mitosis, again, you copy once and divide once. So you're able to go from diploid to diploid. In meiosis, you copy once and divide twice. And that allows you to go from diploid to haploid. Meiosis 1. So here's a diagram of meiosis 1. The purpose of meiosis 1, you will notice that looking at our metaphase right here in the middle, you'll notice that I have my homologous pairs lined up, meaning I have my mom and my dad copy of the same chromosome sitting next to each other in metaphase. So instead of having a single file line, I've got doubles. So the purpose of meiosis 1 is to separate homologous pairs, meaning all of the cells at the end of this are either going to have a mom copy or a dad copy of every chromosome. They don't get both. Meiosis 2, so the second part of meiosis, is about splitting my sister chromatids. So notice that my cells at the end, the cells I made at the end of meiosis 1, so my first round of division, they're going to be haploid because when I look inside, I've only got 1 copy of chromosome 1 in here and 1 copy of chromosome 1 in here, 1 copy of chromosome 2 here, 1 copy of chromosome 2 here. But the chromosomes that I have inside my cells are still duplicated. They're still X-shaped to have those sister chromatids. We remember that one of the ways we can check ourselves, whether it's mitosis or meiosis, is you should never have duplicated chromosomes. So what I'm going to do in meiosis 2 is split those sister chromatids. So this looks exactly like what we did in mitosis, except now we're doing it with a haploid cell. So split those sister chromatids, and you end up with 4 haploid daughter cells at the end with unduplicated chromosomes.